Welcome, everybody. So I'm Daniel Wibberg. I'm here the representative of de.NBI ELIXIR Germany, and I'm happy to see so many people here today for our joint venture together with GHGA. And today we will talk about methylation analysis. And maybe a few words before we start. So I will give you a short introduction to de.NBI ELIXIR Germany and to GHGA. So de.NBI and ELIXIR Germany, is a network that is on the market since 2015. It is responsible for service training cloud computing in the bioinformatics and life science field. We have, for example, 160 tools. We provide more than 60 training courses per year. And we have one of the largest academic cloud systems in background. And you see here on the map, we have 24 partners in Germany that are divided into 8 so-called service centers. For, I would say, almost all topics, there's a specific service center available. We have one for human bioinformatics, crop bioinformatics, microbial bioinformatics, and so on. And on the other side are my colleagues Vanessa and Julia, and they belong to the German Human Genome-Phenome Archive. It is one of the NFDI initiatives here. It is coordinated directly at the DKFZ, and you see here also on the map, there are several partners spread around Germany, and there's a federated network of 6 data hubs that are focusing, like the name said, on human data. And this network represents Germany in several initiatives as GDI or FEGA. And the basic idea of this network is making especially human genomic research easier and more sustainable. And if you want to know more about them, you can go to the web page. I would say that's the same also for de.NBI. You can go to the de.NBI.de web page and check for more information. Last but not least, I would like to introduce our speaker today. It's Nazanin Mirza-Schreiber. She's located in Munich since several years. So therefore, she started also her career, I would say, in Munich as a PhD student and postdoc at the Max Planck Institute in Munich. Her PhD was in the identification of genetic variants involved in dialysis pathogenesis by joint analysis of QTLS and ELIXIR. And since 2018, she, I would say, only moved a few meters, I would say, from the Max Planck to the Helmholtz Institute. And today we are looking forward to your nice talk about methylation analysis. So the stage is yours, Nazanin.
Yes, hello. Thank you for introduction and thank you for having me. I will share my presentation. So welcome to today's seminar, which is the Methylation Analysis, a Practical Guide for Beginners. We will start with an introduction to methylation and how it is measured, and then we will continue with a typical workflow and some tools. And and in the last part, we will cover which kind of analysis we can do with methylation data using some examples. So what is methylation? DNA methylation is a key component of the epigenetic landscape. Epigenetic describes modification of the genome that do not change the DNA sequence, but change the functionality of the genome as like the regulation of gene activity. DNA methylation is a dynamic process that regulates gene expression by inhibiting the binding of transcription factors and RNA polymerases to DNA by adding a methyl group to the C5 to the C5 position of the cytosine to form methylcytosine. So the enzyme that add a methyl group are called DNA methyltransferases. Such modifications are not arbitrary at any cytosine through the genome. They are rather at specific sites. These are regions of a DNA where a cytosine is followed by a guanine nucleotide, and it's called CPG sites. And they occur with a high frequency in genomic positions that are called CPG islands. And they are usually at promoters of genes. Such modifications are driven mainly by our genetic, where a SNP can influence whether specific positions can be methylated or not. And this in turn affect individual gene expression. So, but also environmental factors like smoking, diet, stress, activity, and even aging have a huge impact of our methylation patterns. So methylation plays an important role in the stability and regulation of gene expression and in the development and maintenance of our cellular identity. The dynamic process of DNA methylation makes it very responsive to changes in the environmental conditions, and that have been shown to contribute to various diseases. That's why the examination of such modifications is in focus of studies. But these studies are not straightforward, as we know from GWAS, like genetic studies, where blood is the obvious choice, because methylation is highly cell-specific. So every tissue can have a different methylation pattern. Here's an example where we see the tissue specificity during the human fetal development. So in week 9, which are the blue dots here, the profile seems similar between the different tissues, muscle, pancreas, adrenal, but different from amnion. But until week 22, which are the purple dots, where methylation pattern differences get more obvious. So this indicates how in specific tissue gene expression is controlled. And obviously different tissue have specific genes activated or the degree of gene expression. So regarding methylation analysis, it is not arbitrary which tissue to extract. For example, for neurological diseases, it has been shown that the correlation between blood and brain methylation is less than 10%. So now we know what methylation is and how important the tissue specificity is. How do we measure methylation for our analysis? One of the most commonly used methods is our arrays, methylation arrays, and the commonly used array are manufactured by Illumina. Illumina has several array types for human. The latest is the EPIC2 array, which covers over 930,000 CPG sites. So array makes the use of bisulfite conversion to detect whether specific CPG loci are methylated. So what happens here is that methylated cytosine stays unaffected. I don't know if you can see my mouse here. So where methylated cytosine stays unaffected and unmethylated cytosine is converted to uracil and then thymine. Illumina makes the use of 2 different probe types, which we don't need to go into the detail, but it's important to know that there are 2 different probe types that produce different signals, and we need to take care of this later in your QC to handle these different types separately. So our normalization methods to ensure that the intensities are comparable. So what kind of data do we get from our array analysis? From these measurement techniques, array-based methods, we generate for each sample 2 different so-called IDAT files, one for red and one for the green raw fluorescence intensity measurements. And these files store raw methylated and unmethylated intensities, control probes, which we will need later and we will discuss later for our QC, bead counts, and any kind of manifest index information. Control probes will give us some information with access the information about the quality and the performance of the array for each sample. So another way beside arrays to measure methylation is sequencing. There are different methods as like whole genome bisulfite sequencing, or PacBio, we will concentrate today of Oxford Nanopore technology, here, which uses nanopores embedded in a membrane, and the DNA molecules are threaded through these pores while an electric current is measured. So if there are any methylated CpG sites, the signal changes and is directly detected. There are different models, which is also offered by Oxford Nanopore, like GAPI or Dorado, which can be used for the base calling. So let's go quickly over the steps that we need to apply or need to be applied on the raw sequence data to get the data that we need for our analysis. So as I already mentioned, from the ONT, you will get raw sequence data, which are in form of FAST5 or POT5 files that can be used by Dorade or GAPI for base calling, plus Remora for methylation detection. Then the data need to be aligned by using Minimap2 and aggregated by ModKit, which aggregate per read methylation calls into per CpG site methylation frequencies and outputs them in a bed method format. So how does such a bed method format looks like? So here is an example. So ModKit produces 3 bed method files per sample. Since ONT provides haplotype resolved methylation calls, we will have for each sample 2 bed method files representing haplotype 1 and haplotype 2, and one more for those reads that could not correctly be assigned to either of these haplotypes. So the bed method file contains various information for each CpG site, like the genomic position, the number of reads, the number of the coverage for each CpG site, and it calculates also the fraction per or percentage per modified read, which is similar to the final value that we create when we have array data, which are the beta values. But we will discuss that also later. So another difference to arrays is that we get the exact information whether the modification is a cytosine base with a methyl group or a hydroxy methyl group, which makes a difference because the 5MC or the methyl group is the methylation that we're usually talking about. That is what we expect to suppress genes, like to work as gene silencing. But the 5-hydroxy methylation has probably a different role, which is still not so clear. It is rather linked to gene activation. So it is a nice feature that you would be able to analyze these 2 different types of methylation separately. So arrays, as I said, do not differ between those methylation types, and any methylation that is detected is all just put together as methylation. Another big difference is that the amount of CpG sites that are detected. We have around 28 million CpG sites on our genome, and arrays just cover 3% of them, while ONT reports that they can cover 97% of the CpGs in our genome. So that was now already a lot of information. Now we can get started with our workflow on methylation data. Let's start with arrays. There are different R packages that can be used for processing and QC, like MinFeed. These are some of the common. There are a lot of packages, but these are some commonly used and most popular, like MinFeed, Sesame, Watermelon, or Champ. And to work with the data, you need appropriate annotation packages. These are packages that offers you information for each CpG site, like the genomic position, if it lies in a promoter or in a gene body, or a lot of other information to which gene it is annotated. So, and you need always to take care that you have the correct version. Like for EPIC2, you would need the Illumina human methylation EPIC annotation, which is based on human genome build 19, or for EPIC2, human genome build 38. So, and then you need packages for the analysis of the differential methylation. There are also different packages like Lima or CHAMP, which offers you methods to do your differential methylation analysis per CpG site. Or maybe you're also interested to check whether whole regions are differentially methylated, which would be very interesting if you use a gene expression as a phenotype. These tools are called DMR-CATE, or a function from MinFi is bump hunter. So in contrast to arrays, ONT measures methylation directly on the native DNA sequence. Right? So they are less affected by probe chemistry, design bias, etc. So most variability comes from the biological difference or a sequencing depth rather than from platform-specific constraints. So quality control still matters, but there are actually no specific ONT QC packages for methylation. You can you always use your individual pipelines, or you can also use, like for example, methylkits, which is for bisulfite sequencing, but can be also applied to ONT. So there are also no ONT-specific annotation packages. As I said, ModKit will offer you already genomic position, but if you need further information, you can use any standard genome annotations. For differential methylation analysis on count-based data from the sequencing, you can use the same statistical tools as for bisulfite sequencing, for example, DSS, BSEC, or methylkit. Now let's go step by step through our QC. We will start with arrays. So as we already said, we have IDAT files for each individual. You have 2 IDAT files which need to be which you can read in R using MinFi functions, which will generate you an RG channel set. So now, in order to keep the workflow consistent and avoid mixing different frameworks, we will go with the MinFi functions. But however, for each processing step shown here in MinFi, you have equivalent functions in other methylation packages, like in Sesame or CHAMP, or any other package that you decide to use. So array data are not straightforward to handle as like sequencing. So because arrays are hybridization based, and they are therefore highly sensitive to technical artifacts such as dye-based background fluorescence, probe type differences, and so on. So these technical factors must be corrected to make methylation values comparable across probes and samples. So first of all, we usually calculate our detection p-value, which is indicative of the quality of the signal. So MinFi compares the total signal of methylated and unmethylated sites for each probe to the background signal level, and will give you a p-value. The small p-value is indicative of rather a reliable signal, like p-values smaller than 0.01, and a large p-value generally indicate a poor quality signal. So usually you should exclude all CpG sites with a p-value higher than 0.01. And samples have, if samples have many failed probes, they will have a relatively large mean detection p-value. So here are different samples, and the mean p-value over all CpG sites is calculated. You can plot that to check your data. And if a sample has a pretty high p-value, higher than 0.05, it's recommended to exclude that individual. And of course, you need to exclude CpG sites and samples which have more than 5 to 10% missing values. So in another step, we already excluded samples which are poorly which have a poor quality, and CpG sites with a poor quality. We need also, there are 2 types of probes that we need to exclude from our analysis. So one type are cross-reactive probes, that are probes that have been demonstrated to map to multiple places on the genome, and this can create false signals. So there are lists online that you can download, or there are meanwhile also a package called Max Probes, which you can install, and based on which array type you have, EPIC or the older versions, you can extract all these known cross-reactive probes and exclude them from your further analysis. Also known probes that are SNP affected must be excluded. There are probes that are known to have a common SNP at the CpG site directly or at the single nucleotide extension. So there is also a function in MinPhi, like drop loci with SNPs, which will automatically exclude our known CpG sites with SNPs. So we can ensure that our signal is not due to underlying variants. So now we come to our control probes. There are, for every sample, you will have different control probes on each array that is also in the IDAT files, and then you load in your RG set, and that will give you a hint about the quality of your data. So because control probes capture technical features, staining, extension, hybridization, etc. So you can read out every control probe for each sample and check whether these samples are below a specific threshold or what is recommended. I have put also a paper at the end which you can read more in detail about control probes. It is recommended to take all the control probes on the arrays and run a PCA and check whether any individual is an outlier based on these control probes. And if so, you should exclude this sample because they have a lot of technical artifacts from your further analysis. And what you also need to do is you need the components from the PCA on the control probes, so you can use this later for your downstream analysis to account for your technical artifacts, because EVAs are highly confounded when they are based on arrays. But we will also discuss that later again. So MinPhi offers another step, which is called getQC, that will specify whether a sample should be excluded based on its overall signal intensity. You can always run that and decide whether you want to exclude these samples or not, which from my experience is actually always a good hint to exclude these samples. Further, it is important to do a sex prediction. So we should check whether there are any mismatches between our documented and predicted gender, as methylation is also highly sex specific. So you should take care that this information is correct and matches the profile. So for example, I had already data set where 2 samples were documented as males, but the profile show that these are females. So in our case, it was just wrongly documented, but if you cannot find out where this problem comes from, you should exclude always these samples from your analysis, because it can destroy your results, or maybe investigate these samples more if you cannot find out where this mismatch comes from. As a next step is the background correction, which is also very important. There are different functions with any kind of package you use that removes unwanted background fluorescence noise from the methylated and unmethylated intensity signals. So noise sources can include scanner noise, autofluorescence, non-specific binding, and out-of-band signals. And so you can just put your RG set and use these functions and get rid of these background noises. Another important step is that whenever you have a bulk methylation data, there are always multiple cell types present, like in blood. So blood is a highly heterogeneous tissue, which is composed of multiple immune cell types, like CD4, CD8, and monocytes, etc. And all these immune cell types have also specific methylation patterns. So you don't want your analysis to be confounded, or the difference between case controls that you capture is based on the cell types rather than the phenotype that you're interested in. So there are two different options to get to account for such cell type specificity or cell type compositions. One is the reference-based. So, for example, MinPhi offers a function to account for this different cell type compositions, which you can use on your data. There are reference panels for brain and blood and some other tissue types. And so if you don't have a reference panel due to your specific tissue, if you don't have a reference panel, you can always run a reference-free approach. These are so-called deconvolution methods. There are a lot of different packages that are offered that you can use in R to apply on your methylation data that uses statistical patterns in the data to infer latent components representing the cell types. And this can be later the fractions that you get out of from the reference-based, or the components that you get out from your reference-free approach. You can use that later as covariates in your analysis or in your epigenome-wide association studies. So then the last and one of the most important steps is the normalization for arrays. So to minimize unwanted variation within and between samples, you have to normalize your data. Very important point is that from now on, we will handle our data separately for sex chromosomes and autosomal chromosomes. So because so methylation pattern on the sex chromosome differ fundamentally from those on autosomes due to X chromosome inactivation in females and the hemizygous nature of the X and Y chromosomes in males. And so if you put all together the sex and the autosomal chromosomes, then it can distort your normalization, the variability estimation, and your downstream analysis. So from now on, our analyses are always either just on autosomes, or we handle our sex chromosomes separately. So there are also different methods that you can apply for the normalization, which are also done on the different on the methylated and unmethylated intensities separately to make them comparable. For example, and you have always to check what kind of data you have and which normalization method fits your data best. So there are functions. Here are just two examples, like process Phenome, which you can use when you expect a global methylation difference in your data, such in cancer cells or in non-cancer, in cancer patients or in non-cancer patients, or you have different tissues that you're investigating. So then process Phenome maybe would be the right choice for you, or process Quantile for data where you do not expect a global difference between your samples and you have only one tissue type. So once you did that, you can compare your raw and normalized intensities, and you would expect a clean bimodal. So in best case, you would expect a clean bimodal distribution after normalization of your beta values. and to compare to your raw data values and check whether it looks like as you would expect. There is another function that you can use on your data to check the quality or to have a hint how what to expect from your data. Like you can do a multidimensional scaling analysis where you actually where the function is taking like the 1,000 or 500, you can define that 500 or 1,000 most variable CPG sites in your data and to represent your samples. And that gives you a hint about how the quality of your data is and what to expect. If you see global differences between your case and controls or between the different subgroups, or if you have different tissues to see whether you can, based on the methylation, separate this tissue. So I think it's always a nice way to have a feeling for your data before you do your further analysis. So as a last point, you heard now often beta values. So what are actually these beta values and how are they estimated? So the methylated and unmethylated intensities are used to calculate these beta values, which is the methylation level using the ratio between methylated probe intensities to the sum of methylated and unmethylated intensities. It ranges between 0 and 1, for 0 being unmethylated and 1 being fully methylated. And the beta values are optimal for visualization because they are very intuitive. So you know actually, okay, 1 is methylated, 100% methylated, and 0 not. And but for analysis, it is recommended to use M values, which is the log2 ratio of the intensities between methylated and unmethylated probes. Where here also a positive value means more methylated and a negative value means less methylated. So, but why M values? Beta values, it has been so beta values tend to have a high variance heterogeneity. So Pandu and colleagues show that the standard deviation is highly compressed at the lower and higher levels, and which is not the case in M value. So the standard deviation is almost constant across the entire methylation range, which is more appropriate for the assumption of statistical models that are used for microarrays. So what we do, we use beta values for representing our data and visualizing our methylation values, but we use M values from array data to run our analysis. So now we did our array QC. Let's go quickly to our Oxford Nanopore QC. Here we don't have so many steps as for arrays, since ONT or sequencing data is the actual biological readout of the sequence, and as said, you don't have all this technical bias in your data. So important is to exclude CPG sites with a low coverage, less than 10, and exclude CPG sites that are missing in more than 5 to 10% of your samples. Another point is, since UNTA offers a huge amount of CPG sites, like over millions and millions, it would make sense to exclude CPG sites that don't show any variance between your samples, because you have always to consider that you will have a problem regarding multiple testing. Of course, you have also to estimate cell type compositions. There are also count-based models like Selfie and Selfish and some others, which you can use on your account data to estimate your cell type composition. Another step is to exclude samples with low mean coverage, less than 10. So here's a very extreme example where a lot of the individuals had a mean, so you calculate the mean over the coverage for each sample, and if this means is less than 10, you would exclude these samples from further analysis because the data is not reliable. So now we spend a lot of time doing our QC. Let's start with some analysis. So let's assume we have clean data, we have the right tissue, we have all our covariates that we need to investigate our phenotype of interest. So similar to genome-wide association studies, we can perform epigenome-wide association studies to find whether there are any differentially methylated sites or regions between our cases and controls. So I think the principles of EVAS is clear. We check for the association of each CPG site to our phenotype between our case and controls, and or our disease status, whatever. So important is for EVAs to always keep in mind that they have high inflation due to many possible confounding factors. You need to take all possible factors into account: age, sex, smoking, any kind of information. You should use that as correction factors. And of course, the PCAs from your control probes, then your cell type composition, any batch information that you have, you should take that to correct your analysis. So once you did that, you can run your association study and hope to find some significant CPG sites that are associated to your phenotype, using functions for if you have array data, you can use lemma, or if you have count data, you can use DSS or BSEC. So once you've done that, you hope to find some CPG sites that show a different methylation pattern in cases compared to your controls. Once you did that, maybe you are interested to see whether you have any differentially methylated regions. So where the average methylation level is significantly different between your cases and controls. That is always very interesting if you have like expression data where you can check whether there are differentially methylated regions that are associated to your expression of a gene. There are also different tools that offers functions to run such analysis, like for arrays DMRcade or bump hunter, which is a function from Menfi, and there are also other from Champ, etc. There are also other functions that you can use, and also count-based methods like DMR-seq or DCC that you can use to investigate whether you have differentially methylated regions. So, but there is much more that we can examine using our data, like calculating the epigenetic age. So one of the first methylation age predictors that was developed was from Harvard, which allows one to estimate the DNA methylation age of most tissue. So meanwhile, there are few other age or phenotypic predictors like phenoage or Grimm age that predict health status or morbidity or mortality risk, or MEAT that predicts age for skeletal muscle. I think it's always a nice add-on when you can use your methylation data to check whether you see a difference in your cases and controls. Does the cases maybe show a higher age acceleration compared to controls, or is the morbidity risk for your cases higher than in your control? So I think it's always a good add-on to your analysis and gives you good information about your data. So now let's go to our last topic, which are methylation as diagnostic biomarkers, so-called episignatures. So the diagnosis of Mendelian genetic disorders remain a challenge. So despite advancement in genomic sequencing, there is always the need for accurate biomarkers, which also maybe are cheaper than whole genome or whole exome sequencing. So there are genetic syndromes that can have a very unique genomic DNA methylation pattern. These are called episignatures. So some pathogenic mutations have a genome-wide impact on DNA methylation. So these patterns represent a very early stage during embryonic development. So, and are present in various tissues. So that's why they are also present in blood and can be measured in blood, which is always the easiest way to collect data. So it is known that signatures are very specific to the diseases, which can be used to resolve ambiguous clinical cases and to identify undiagnosed cases. So there are meanwhile for many Mendelian disorders episignatures, but how do we define such an episignature? So usually you need a good training set, you need a test set, you need a validation set. You start with running an EVAS and to find the most variable and effective CPG sites in your data, which you need to verify using different clustering methods. And then you can use this CPG site to train on a supervised model. So let's go quickly over an example. So our group could identify de novo mutations in the gene KMT2B in patients with sporadic childhood onset dystonia. Dystonia is a highly heterogeneous neurological disorder that has at least over 70 different subtypes. It is a hyperkinetic motor disorder that disables posture, movements, and often show intellectual disabilities in the individuals. So as we knew that this gene is involved in the epigenetic machinery, since it is lies in histone methyltransferases, we tested if we can create such an episignature for KMT2B. So first we created a test and training set, and we had also an validation set. So we started in our test set to run an EVAS and chose an effective subset of CPG sites that was significant and had at least an absolute log-fold change higher than 1. Then we did a hierarchical clustering of the specific signature to separate cases from controls in our test set, and we saw that it could perfectly separate the cases from the controls. We could observe a high hypermethylation in the KMT2B patients compared to non-KMT2B patients. And since this worked very well, we used these CPG sites to train a support vector machine. So what does the support vector machine do? It generates scores ranging from 0 to 1 to representing the confidence of a prediction for each class being a KMT2B case or not. So our model was able to successfully predict disease status and evaluate variants of uncertain significance from multiple batches. So as different studies meanwhile showed, these episignatures can be very powerful biomarkers. So yeah, with that, I would like to come to an end. I think I'm already over the time. So in summary, we went through methylation design, the quality, the different measurement types. Then we talked about how confounding methylation data can be, the tissue specificity. We talked about EVAs on epigenetic clocks, what we can use them for, and methylation as diagnostic biomarker. I have put here a table with some papers and reviews for further reading. I think they are very helpful if you start your own methylation analysis. And here's also a table attached where you have some different functions to start with some information to start your analysis. And I would like also to acknowledge my group leader, Konrad Oexler, and our director, Juliane Winkelmann, my colleagues, Barbara Schaumeyer, Michael Zech, and Philipp Hara, and also Bertram Müller-Mosak. So thank you very much for your attention. I hope it was not too much, and I'm happy to take your questions.
Yeah, thank you, Nazanin, for this nice talk about methylation analysis. Have a nice day and see you next time.
Also, best wishes. Thank you.